Okay, so most of the lectures this week, there is a hour and a quarter long lecture, then an hour and a quarter long hands-on session. And that worked, I think, for the stuff we've done so far. This, just based on the nature of the material, I think is much better to sort of intersperse the exercises throughout. It's just there's a bunch of things that are not hard. They look complicated. You try them, and you see exactly what's going on. So I wrestled with how to do this, and I try to separate them, but I think it's best to just do it this way. So it'll be nice, because we're at the end of the day, it's hard to sit for an hour, hour and a half, look at something and focus. So think of this as like a few exercises that sort of wake you up along the way and catch up. Okay, so an overview. The main question is, given a raw sequence of RNA, can we predict how it will fold? And let me just mention that the expert in the room, probably sitting over there, David. Well, I know I've taught a graduate. I've not published a paper in this. I have taught a graduate course. I've advised a master's thesis. When I taught the graduate course, I was asked to do something math-bio related. And my colleague, Elena Dimitrova, had recently done a discrete model in class. So I knew of this book, which is a colleague of mine had recently just published this book, Christian Reddy's Combinatorial Computational Biology. He's a mathematician who I was in Los Alamos with about 11 years ago. And so I said, well, I want to learn this stuff anyways. I might as well learn it and teach it and be a step ahead of the class. And so I had 11 grad students, and I said, I'm not me teaching this to you as students. We're in it together. There's statistics in here. There is optimization. There is combinatorics. There is some really hardcore analysis. And I had students from different backgrounds. So I said, let's figure this out together. So it was a 985 class where we came, and there was a few assignments throughout the semester, and everyone got an A because everyone participated and did fine. But it was a fun learning experience, and we got some interesting ideas out of it. So I've had that experience, and one of my students continued on that little project we had at the end and make it into his master's thesis. So that's my experience with this, and we have also hired Svetlana Postanovic, who was a postdoc of Christine Heitsch, who you saw some of her pictures, and her replacement for the postdoc is David up there. So David has spent the last 2 years at Georgia Tech studying some of these questions. So if you have questions, talk to him as well. Talk to me. Talk to the biologists in the room about the biological details. But here's the question: how can we predict how it will fold? There are two approaches to this problem, from what I can tell. There are one approach is a dynamic program, which is a recursive algorithm, where you figure out, given a folded structure, how much free energy it has. So you give an energy score to every structure, and it turns out that the most likely structures, the most observed structures, tend to be those where the free energy is minimized, or really close to minimized. And I'll talk about what that means in a second. But the other approach, one more thing. So this free energy, this score is computed recursively using dynamic programming. And that's what I'm going to do today. So today is going to be this, an energy minimization approach to RNA folding. Tomorrow, I will show you, for who is interested, in formal language theory. This comes out of computer science, comes out of linguistics, using a formal grammar to algorithmically generate secondary structures. So this consists of production rules that convert symbols, temporary symbols, into other temporary symbols and other permanent symbols. And eventually, you have only permanent symbols left, and you have a word. So there are syntactical rules. This is why it says formal language theory. This was developed. I don't want to say developed, but it was really popularized by Noam Chomsky, a professor at MIT. And Charles Darwin in the 1850s wrote in his book, Origin of the Species, that there is a really interesting similarity between the evolution of the species and the evolution of languages. There's a lot of common syntactical underlying structural similarities in both of those. And this is before anyone had any idea that there was this language of evolution. So he noticed this decades before anyone discovered DNA. And so once DNA was discovered, then people started using linguistic approaches to study DNA sequences. And so DNA was, so it was cracked in, I want to say, the late 50s, is that correct? And then it wasn't until the 1980s that people started using these formal language theories to study DNA and RNA. OK, so this will be the part 2 tomorrow. It's just a much different way to do it. This is a deterministic. It will pop out a solution. Like, this is the minimal free energy structure. Could be 2 or 3. But in general, with all the parameters that people nowadays put in, it's going to be 1. This is everything. This is sort of like what we saw earlier today. You're going to have a lot of possibilities with different probabilities. There's randomness in here. So you are going to have a most likely structure, but you're going to have some others, second and third place, that could arise as well. And realistically, we see an RNA molecule fold in multiple ways. So an algorithm that just gives you a deterministic answer is not going to be entirely accurate. So a perfect algorithm would be a stochastic one that gives you a distribution, or that gives you probabilities on all the structures that matches what we see. Now, that is not going to be realistic, but that would be the gold standard. And both of these perform about equally well, last I've seen in the literature, anyways. OK, so today we will do this. So yeah, if we assign probabilities to the rules, then the most likely structure is the one that occurs with the highest probability. So before we start, I will establish some mathematical notation, just so we have a framework and some ground rules, so we know our boundaries and we know what we're talking about. Yeah?
Quick question. The formal language, you're only doing the secondary structures?
As opposed to tertiary structures, yes.
Okay.
I had to think, because we had this. There's a discrepancy about secondary structures, which I will talk about in a few minutes. OK, so to each base, so the base pair is like A and U. A base is a single nucleotide. We associate a vertex. So I don't like the word nucleotide because it's too many syllables. So I will say base or vertex. Each base pair, there'll be an edge. Okay, and that denotes a bond, a chemical bond. So we arrange our vertices, our bases, linearly in a line. And the arc diagram of an RNA folding, a structure like this that folds, we arrange the vertices in a line. And the bonds are represented by edges or arcs that we usually draw on the upper half plane. So we have a bunch of vertices. And that might bond to this. That might bond to this. Something like that. Let's put one more in there. OK. So there are several natural combinatorial models we can associate with RNA strands. And I think a picture is worth 1,000 words. So here are 4,000 words right here. You can view it like this. This comes from Christian Reddy's books. You can view it as a, so I think this is clear. This is like a, what was that called? What was the?
tRNA.
What was the?
Cloverleaf. Yeah.
Cloverleaf. This is a cloverleaf diagram. Yep. This is probably the next easiest one to see. You can, if you just stretch it out and draw arcs for those bonds, you get, this is the arc diagram. This is a point bracket notation where you can see what's going on. The beginning of the arc, you put an open bracket. The end, you put a closed bracket. And notice how this completely determines this, assuming you don't have crossings. And then you can also, instead of doing it this way, you can look at, this is called a Motzkin path. Every time you have a bracket or you're starting an arc, you go up. Every time you have an isolated base, you go across. Every time you end, you go down. So just different ways to visualize things, and they have their advantages. Yeah?
There will never be crossings?
Not yet.
Let's not go there.
Oh, I'll go there, but I'll mention it. So this book is called Pseudonauts. That's where you have crossings. OK. So what I want you to do now is here is an RNA sequence. I want you to draw an arc diagram. And I want you to draw the arc diagram. I want you to do the point brackets underneath. And I want you to do the path. So I'm going to give you a series of exercises, keep you awake a little bit, and also so you follow. Otherwise, when I keep giving you these definitions and examples, it's going to be, it's easy to lose you. So let's see if, so I'll go ahead and do this right now. And if you don't finish one of these exercises when I move on, that's fine. Anybody need paper? Not to guilt trip you, but everyone knows what it's like to teach a calculus class and say something like, okay, now you try this on your own, or work in groups, and one person or two people just sit there. So I'm not looking at you, so I'm not singling anyone out, because I actually don't know if there is anyone doing that, but I didn't want to put anyone on the spot, so I'll look back now. Okay. So yeah, just draw the sequence, put numbers underneath it, and then put brackets and. This sort of represents how this thing might be laying down. And so these are the bonds. You can think of this as like Velcro. Three G's, A, 1, 2, 3, 4, 5, 6, 7 G's. Yeah. Everyone has a sequence down. I'll show you that.
She's here already finished. So you can go. So yeah, but sorry, I need a computer science approach. You talk about the chemistry one. You need the diagram. If you go with that bottom one.
And if anybody finishes early and you're waiting around, I encourage you to go to this website. This is Etern, I don't know how to pronounce it, eterna.cmu.edu. This is a game you can play where you are playing around with nucleotides and you see how they fold. So don't do this like you would with Tetris or Candy Crush. I mean, don't actually do this during the lecture, but if you finish early, go here and explore it. What? Oh, you want to see the next slide? Yeah. Okay, so I'm going to continue. If you haven't finished, that's fine. I just want you to get used to playing around with some of these. So you should notice that your arc diagram has no crossings. These things are going to, you have these arcs here. So what you will, this roughly looks like is you'll have a, you'll have something like this and something like this, right? Along your backbone. Okay, so formally, if I want to make this formal, I could say two arcs are crossing. So what does it mean for two arcs to cross? That means, so let's call this I1, I2. No, sorry, I1, J1, I2, I1. Yeah, I2, J2. So formally, I say two arcs are crossing if I1 is less than I2, which is less than J1, which is less than J2. And an arc diagram is we say an arc diagram is non-crossing if it has no crossing arcs. So now I have one more. So an arc diagram, an RNA structure is typically called, unfortunately, a non-crossing RNA structure is called a secondary structure. And the reason why that is unfortunate is because this thing here is, so the primary structure is just the raw sequence of bases. Just that sequence there. The secondary structure is just the raw sequence of edges, or pairs. So this, so given those two things, you can't tell if there's any crossings like this. So so I would call this a secondary structure, but historically, this is not called a secondary structure. Historically, for years, only structures that had non-crossing diagrams were studied. So secondary structure was synonymous with non-crossing diagram. Later, these things arose. People saw these once in a while, and they called them pseudoknots. But the word secondary structure had already meant non-crossing. So it's unfortunate that there's a little bit of ambiguity, but the literature seems to agree that secondary structure means non-crossing. So I'm going to call this a pseudoknot.
These are kind of, you have to do with the third dimension, because you glue 2 points together, right? And then part of that loop is going to have to go over.
Yeah. So watch, see that diagram? I'm going to give you the same sequence,
Okay.
but watch that. So this is the same sequence, but this is folded differently. So underneath that, what you did, I want you to try to do the same thing. Draw the arc diagram. Draw the point bracket notation. Not all of these things will work. And I want you to sort of realize, so you will be able to write this point bracket notation. I don't want you to see what goes wrong with 2 and 3. Something will, but I want you to just notice it yourself. The arc diagram should work. And the previous slide had, notice what happened. Like previous slide, these snap together and those snap together. This slide, those snap together. So you'll notice that you can create a point bracket notation for this. But then if you give that point bracket notation to someone else, say reconstruct it, you might not get the pseudoknot. You might get they might reconstruct a secondary structure. So once you get into pseudoknots, you don't have this one-to-one correspondence between structures and notations. Now you have a multiple-to-one structure. A many-to-one map.
Are you saying that mathematicians wouldn't call that one a primary and that a secondary?
No, the primary is the string of, is just the pure sequence.
No, I understand what the biologist
Okay.
But were you saying that mathematicians wouldn't have wanted to call that primary and
No, this has always been secondary. Yeah, and for a long time, people only, this is a secondary structure.
I think mathematicians would have called it crossing and non-crossing.
Right, yeah. Right. So, okay, so what is in the literature? So what is, I don't know if I'm going to be able to draw this, but this thing here. So let's. So this here. Let's suppose this 3' end were a little longer. Just knowing the sequence and knowing the pairs does not tell you whether this longer end is going to tangle in and not in and out of there. It doesn't tell us that this end, this is 10 longer, it could loop up into here. That is not given by just the sequence of bases and the sequence of pairs. So that little extra knottiness is part of the tertiary structure. Whereas the secondary structure is just the base pairs. Unfortunately, for, I want to say decades, people never even considered this type of structure. They're not as common as the non-crossing ones. So people just, when they said secondary structure, it was synonymous with non-crossing structure. So they meant exactly the same thing. And then someone said, well, what about pseudoknots? We see some of these. And now, well, secondary structure was already taken. So they call it something else. And so there is a little ambiguity. OK, so this is an open-ended question. What goes wrong if there are no crossing arcs? Hopefully you've, instead of answering this, I'll just say, I'll let you discover it. And so you'll notice that you can do the point bracket thing, but nowhere in there does it say that it's a pseudoknot. So you've lost information. So these things are not really that helpful for encoding pseudoknots. OK, so here's a definition I want to give you. An arc diagram like this is k non-crossing if there is no set of k mutually crossing arcs. So so the diagram you drew for this thing, it will look roughly like, I'm going to make this bigger. It should look something like this. This is 3 non-crossing. Because if I take, there are no set of 3 arcs such that any 2 of those 3 arcs cross. Like if right here, this is a pair of crossing arcs, 2 arcs that cross. If I throw in any other third arc, it's going to either be it's not going to cross both of these. So this is a 3 non-crossing structure. If I were to add that arc, now this would be not 3 non-crossing. It would be 4 non-crossing, because here's a triple of arcs that crosses, but there's no 4 tuple of arcs. OK, so here I've let's just talk our way through this one. So I've taken, is this the same thing as this is a slightly different, yeah, it's a different one. And let's draw the arc diagram. Now I'm not going to do every single base. I'm just going to roughly sketch what it looks like. So here we start out, and this is going to be a loop that's going to. So I'm going to have 3 of these things here. And now, let me ignore this for now. This is going to be a loop like this. Like that, right? And then this loop here is going to start from inside here and go out there, right? So this is now a pseudoknot. It is 3 non-crossing. Does everyone agree with that? You can find 2 arcs that mutually cross. You can't find 3 that mutually cross. Is that clear what I mean by that? Does anyone ask about? OK. So my next question is, what if the first G bonds with the C directly below it? So this is 3 non-crossing. What if this thing folds down and bonds here? How would that change? So let's do that. So now we have the first guy is bonding with, where is that C? That C is inside of here. So is that going to change the crossing number? is it still 3 non-crossing? I think so, right? Let's see, you can point out that's going to still be 3 non-crossing. So here's the exercise I want you to do. This is going to test your right brain, left, whichever one is the one that we are weakest on. I want you to draw a picture. So here, I've tried to draw a picture of this RNA strand lying down in the plane, and I've pictured these things as like Velcro, and this snaps together, this snaps together, this snaps together. So it's planar in the sense that none of these bonds are really crossing. These chemical bonds are short. I don't want to actually artistically draw this as a big arc. So I want you to draw a sequence like this. You don't actually fill in but the basis. Just draw a line and try to make it 4 non-crossing. See if you can do that. So here's how I want you to start. So suppose you have an RNA strand and something like this, and I don't know, maybe connect these together, something like this. Is that going to be 4 non-crossing or 3 non-crossing? Try to do it. You can, but I want you to. One thing that scientists have noticed is that most of the pseudoknots that are observed are at most 3 non-crossing. So mathematically you could create a 6 non-crossing structure. Physically, that's not realistic. So I want you to try. I want you to try a few things, and I want you to see if you can come up with, maybe this is, I don't know. But see if you can come up with a little squiggle, like a little cartoon, that actually is a 4 non-crossing diagram. I'll give you a minute to do that. And there'll be some trial and error involved in this. And what I don't want you to do is I don't want you to say, oh, well, I need to do that, add. I don't want that. Now, maybe you can do it if you could visualize, oh, I could have done it if I could bend this thing in and then snap that together. But that should help motivate why these things are hard to make with more than 3 non-crossing. I should offer a reward for I wish I had candy or something. I could offer a reward for whoever comes up with it first. Did I actually make one accidentally? I hope not. No, I don't think no, that's not going to be. Yeah, this is certainly not a. No, it's just as big as. Does anyone have one? You want to come up here and draw it? All right. Oh. No, I want you to draw it like a little cartoon strand here and snap these pieces together. So I want you to actually start with a realistic picture. Oh. Okay. I want you to draw the embedding. So how is this? So let's see. I want like this. I'm going to try along with you as well. So red, and I want a blue. So I want the black band to go from here to partway. This is. It's hard. I know I've done it before, but it's tricky. Let me see. So that might start. So maybe I want something like. Yep. Like that. That's going to be.
No.
Oh.
This one. That's this one.
Yeah, but this doesn't have any crossings.
You said 4 non-crossing.
Yeah, 4 non-crossing.
1, 2, 3, 4 non-crossing.
Yeah, 4 non-crossing means it is. Yeah, but it also has to be 3, but not 3 non-crossing. So there has to be some triple of arcs that all mutually intersects. Okay, so I'll let you play around with that one. Does anyone have it? We can move on. I mean, I think the exercise is to see how hard it is physically to have a 4 non-crossing or 5 non-crossing. I thought I could do it here, but it's a harder exercise than. Okay, so so draw a picture of a folded RNA strand. Like, yeah, that was the last exercise. Okay, so let me give you some definitions, some parameters. The length of an arc, ij. So if we're going from vertex i to vertex j, the length of the arc is just I minus j. So it's, if you draw the non-tridimensional diagram, it's the length, the number of vertices, j minus I. And sometimes we'll call an arc of length k a k-arc. Now this is not the physical length of the bond. This is just the number of nucleotides that it jumps. A stack, also called a stem or a sequence, is a stem or a helix is a sequence of nested arcs. So think of it like a rainbow here. That's in terms of folded structures. If you looked at those cloverleaf diagrams, the motivation for calling it that was, I'm not going to be able to. There we go. Yeah, so you can see that these things are called stacks, or a helix, or a stem. Any of these things are used. So maybe this is the 5 prime. This is maybe the 3 prime. And then maximal such sigma is its size. So I will say this is the size of this stem is size 5. Well, there's also a 3 stem in there, but the size I'm going to say is the largest maximal size. So here's mathematically what this is. When I say mathematically, I'm really saying, not just to please us mathematicians. If you want to program this into a computer, you need to have a language, a simple system you can tell the computer what is the length. So this is how, algorithmically, you would tell a computer what a stack is. So for thermodynamical reasons, there are several key features of interest to us. Two things in particular. The minimum loop size, or the minimum arc length, that parameter is called lambda. That is the, and also the minimum stack size. So it is common to assume that sigma is equal to 2, and lambda is 3 or 4. And let me try to motivate it with a picture again. I'm going to move over here and erase some things. So we are interested, biologists are interested in understanding how RNA, in the end state of how an RNA structure folds. So it is absolutely possible that an RNA structure might fold like this, and with a single bond, but it's not going to end up that way. This is weak. So this thing's going to be moving in the cell, and something's going to knock this, and if this is a single bond, it's probably going to break apart. So think of this as Velcro. Once it folds and it clasps together and bonds, it's stable, it's not going to break apart. But temporarily, in the process, this might be an intermediate state. Think of the bonds as glue. So realistically, if there is a, let's see if I can do this. So let's suppose something folds like this. This is a single bond. This is not going to persist. It's going to get bumped. It's going to break. And this is going to turn into a bigger loop. So this might be an intermediate stage, but this is not going to be a final stage. So it's typical to assume that the minimum stack size is at least 2. So if we had 2 bonds there, this is strong enough that it's not going anywhere. If we only have one piece of Velcro, it's loose enough it's going to broken up. Realistically, there's usually more, but this is like the minimum parameter that we have, sigma equals 2. Now the arc length, usually the arc length is set to be 4, but sometimes I see it in papers as 3. That is just saying if you have a RNA strand, think of this as like a piece of rope. You can't just bend it arbitrarily. It's thick, it's rigid. While it is, so let's see if I can draw this. It is impossible, like physically impossible, for adjacent nucleotides to actually bond. Like it is possible, I think, for things that are two apart to bond. But it takes so much energy to bend that. It's like trying to close it's like, a box you can't close. It's like trying to bend it over and close it with a single piece of Scotch tape. You can do it, but it's the moment you walk away and let go, it's going to break apart. So these things could physically bond, but they don't. And things 3 away, these things could bond, but it's very rare. I think so while this could bond, it's usually not going to persist. Usually they say the minimum arc length is 4. So anything else is too tight of a loop, and it's might exist temporarily, but then it gets bumped and it breaks apart. So we typically have these parameters for physical reasons. So here are some mathematical questions. Let me just throw these out. People have studied how can we enumerate the number of structures with certain parameters. How many things like this are there? It's more interesting asymptotically, maybe not exact formulas, but do the number of things, does this grow exponentially, quadratically, logarithmically, things like that. So this may require asymptotic analysis, a lot of complex analysis applied to combinatorics and generating functions. Those are the tools that people use. How can we uniformly generate an RNA structure? What if I have I want to say, give me an arbitrary, I have a sequence of 100 bases, and I want to say, give me an arbitrary folded structure. How do I generate, pick one out of a hat. How do I do that? How do I generate these things so this arises as much as the other one? People have studied that. What is the distribution of certain motifs? I know my colleague Svetlana Pozanovic, with David's postdoc advisor, I don't know if you are looking at this as well, have asked questions like, given a random RNA structure, how many hairpin loops? This is called a hairpin loop. How many of these things are there? Over all possible RNA structures, folds, what is the distribution of hairpin loops? And they have determined that they are normally distributive. How many stacks are there? How many multi-branch loops are there? These quantities are normally distributive. That just means bell curve. They have used a stochastic grammar to generate these structures. And using that algorithm, they say the number of hairpin loops is normally distributive. So using their algorithm. Now, experimentally, it's not exactly normally distributed. The distribution is a little bit tilted to one side. So it might look like it's asymmetric, might look like that. And I was talking to Rob about this yesterday, and part of the reason is because the RNA strand just doesn't appear. It gets pushed out, think of like a spaghetti maker like this, and so one end of it is going to bond quick before the other end even gets out. So you're going to have more loops on the left-hand side than the right-hand side, for example. So that's why these models are not perfect. There's no way, to capture that in a stochastic grammar. But it's a little slight discrepancy that arises just because of the biology. And I got a colleague in Denmark who's studied the topology of one of these structures. If you know what that means, great. If you don't worry about it. So they study this thing as a surface and look at the genus and break apart the surface that it's on recursively and use that to generate these structures.